Okay guys, we're going to start on chapter 17. It's going to be on the non-specific, also called the innate immune system host defenses. Let me get my pen here real quick and we'll get started. The innate defenses is going to give us resistance to most pathogens of any type. Now, there is a type of this which is species resistance. You may recall that I told you that there are certain diseases we just can't get, like distemper from our dogs, because our cells do not have the proper receptors, and that's what we would call species resistance. The reason these diseases don't attack us is because we're just not configured correct for those guys to have their pathogenic processes work, which is all we've said right there. However, we do not have innate resistance to a lot of different pathogens insofar as this species resistance. Okay, as far as our immune system, our main line of defense, we have the innate immunity, which we're going to talk about in this PowerPoint. And we also have adaptive immunity, which we'll talk about in chapter 18. Okay, as far as innate immunity, there are two lines of defenses. First is simply the physical barriers. The skin and the mucosal membrane are actual barriers keeping the bad guys out. In addition to these guys, we have some cells and chemicals in our blood and different processes going on which will be triggered by any bad guy that attempts to enter into our body. Now, these guys are not specific. That's what we're going to save this for the adaptive. This works on any bad guy that comes through here, and we'll talk about it in detail right now coming up. So, structures, chemicals, processes, skin and mucous membrane, which are in the respiratory, GI, urinary, and reproductive. Well, those are really one system. Physical barriers. We also, in addition to this, we have some mechanical defenses. And the two main ones that we'll talk about, this is something that hopefully you guys remember back from Anatomy and Physiology 1, is that we are constantly shedding the outer layer of the epidermis, and that layer may contain bacteria, and when the skin flakes off, the bacteria is carried away with it. In addition to which, we have the mucociliary escalator, which, if you recall, in the respiratory system, this is from Anatomy and Physiology 2, we have the cilia on the pseudostratified columnar epithelium, and these guys are going to be moving up from the lungs toward the pharynx. Now, recall that there's mucus in here in which the bacteria get trapped, and then the cilia are going to wave and actually push them up to the pharynx, where when we characteristically clear our throat, we move it from the respiratory to the GI. It goes over into the esophagus, where it's swallowed, goes to the stomach, and the bacteria get an acid bath. So there's our physical barriers, both the skin itself, which remember is salty, it's dry, and we have these interlocking plates or cells rather that prevent the entrance of the bad guys into the inside of our bodies. Epidermis, tightly packed, shedding we talked about. And remember, we also have those dendritic cells right there at the very bottom of the epidermis or at the junction. And these guys are phagocytes that will swallow these cells up and destroy them, in addition to which they will migrate to a lymph node nearby and show this to the specific immune system. As far as the dermis, those collagen fibers just help us keep from having the skin break, and if the skin breaks, then we have lost the integrity of the barrier wall. We talked about this, or at least I have, in the A&P classes when we talk about the skin integumentary system. We also make these antimicrobial peptides. It's kind of like a homemade antibiotic called defensins. The cells of the dermis are going to secrete these guys. Perspiration secreted by the sweat glands. We got the salt, which will take care of a lot of them. And there's yet another homemade antibiotic called dermacidins. So these two, the defensins and dermacidins, there really should be an I in there, guys, are going to punch holes in the bacterial cell walls and causing them to lyse. Lysozyme, which is yet a third. This is something that we have in tears as well. That helps to keep the bacteria off the surface of our eyes. Will also destroy the walls. So we have antibiotics that we manufacture, antibacterial molecules that are going to attack and destroy some of the bacteria that are there. There's a scanning electron microscope of the skin with the cells dry, dead, salty. Mucous membranes, there's the cilia we were talking about. And basically, if you recall, the mucous membrane is basically stratified squamous epithelium, only it's non-keratinized like the skin is. This is going to line, and it's going to be a barrier, nose, mouth, lungs, urinary, digestive tracts, places where we have openings to the outside world where potentially bad guys could get in. Mucous membranes, the epithelial cells, tight junctions. The mucus we've talked about, sticky substance, kind of tar-like. Covers, protects those cell layers, and also traps the bad guys, and also some of the just dust and junk that we breathe in. And of course, these guys also have those antimicrobial peptides in them. Endothelial cells lining the urogenital, blood vessels, lymphatics. This is just a specific type of epithelium. Hopefully you guys have learned this already, but if not, here it is. Tightly packed barrier cells. The endothelia of the blood-brain barrier, as an example, is going to protect the central nervous system, and it's going to be very selective in the type of things that it allows to transit into the brain. Okay. Mechanical defenses, shedding, expulsion, mucociliary escalator, feces, and actually in our urinary system too. The bladder is sterile. Most of the urethra is where you start to pick up bacteria, but the flushing action of the urination is going to wash out a lot of those. And of course, we've got our tears that are constantly flowing across the surface of our eye, and we've already said there's lysozyme in there to help destroy bacteria. And then we blink. The reason we blink is not, somewhat for light, but the main reason is to act as like a windshield wiper and to clean off the surface of the eye. Microbiome, this is our normal flora. These guys are there and they keep really bad guys out because they basically crowd them out. They take whatever nutrients they take them out. Some of these guys actually produce their own antibiotics. Yes, bacteria make antibiotics against other bacteria. Some of them do. These guys are also, their very presence is going to stimulate that second line of innate defense, the cells and the processes and the chemicals. And of course, some of these guys, the ones that line our colon, actually make vitamins for us. Some of the B vitamins and vitamin K, which we need to clot our blood. Okay, as far as our defenses, non-specific, cellular barriers, we talked about that. The mechanical barriers, we've talked about that. And of course, our normal flora microbiome is the resident bacteria. And there are resident bacteria in our mouth, skin, the upper respiratory tract, which is anything above the pharynx, the GI tract, and also the genital urinary tract. And these guys help keep the really bad guys away from us. Okay, chemical defense, lysozyme breaks NAG/NAM. Urine also has a low pH, which means it's acidic. Lactoferrin sequesters iron. Bacteria need iron in order to thrive, and we tend to hoard that iron in order to keep it away from them. Cerumen, earwax in our ears, is to entrap bad guys or small insects, whatever can get in there. The surfactant in the lungs has some antibacterial properties. Antimicrobial peptides, AMPs, broad spectrum antimicrobial properties. Some are routinely produced, like the lysozyme. Some are produced by the presence of pathogens, and that's usually inside the body. The defensins, we talked about epithelial cells. These guys can also be secreted inside our host cells, damage the plasma membranes. Bacteriocins are produced exogenously by certain members of the normal flora within the GI tract. Remember I told you that the certain bacteria make antibiotics against other bacteria, and there it is. Now this is something that you really want to spend a little bit of time with, guys, because this is one of those things that folks find a little bit hard, because it's not something you're going to just pick up with one hearing. Complement, we have a pathway of complement. We have an alternative pathway, and we have the classical pathway, and we have the lectin pathway. So complement is going to be like a series of dominoes. It's a cascade. C1, well, let's just go with the classical. If one of our antibodies binds to a bad guy of some type, that will cause C1 to interact with C2 and C4. These are all different molecules in this system. This will interact on C3, and this will cause C3 to split into a C3A and a C3B. These guys are going to be, we'll talk about them in a minute. Let's just keep going. C3B is going to interact on C5, and it's going to cause it to split. C5B is going to interact with C6, 7, 8, and 9, and these guys are going to form a ring structure that is going to basically punch a hole in the bacteria and cause it to lyse. That's our complement. Complement is very important for our immune defenses. So there's one way you can get the complement cascade going with antibody antigen complex. The lectin mannose binding lectin. Mannose is a sugar that's often associated with the glycocalyx of the bacteria. That will start, but instead of interacting on C2, it'll interact directly on C2, C4. The alternative pathway, and this is spontaneous, C3 is going to spontaneously at some point break into C3A and C3B, and it just starts at different points in there, guys. So you need to know all of those. Lysis, these guys are, that's right, I better tell you about these. These guys are going to act as attractants, chemotactic attractants for the white cells. These are also going to act as pyrogens and cause us to spike a temperature. And that temperature, our immune system works better at higher temperatures, and the bacteria work less well. That's why we get a fever. So, inflammation, we're going to open up or cause the blood vessels to dilate, and that's going to make it possible for the white cells to leave the blood and get into the tissues where the problem is. Classical pathway, there's the antigen-antibody complex. Alternative pathways, endotoxin, glycoproteins, spontaneous, C3b, the lectin, there's the mannose and the lectins. And all of these things are basically going to do the same thing. C3 splits into C3a, C3b. Going to cause C5 convertase. That's just an enzyme to split C5. C5b is going down here, and C5a is going over there. They've actually got these things kind of labeled a little bit odd. Classical pathway complement. There's the membrane attack complex. There's a lot of C9s. You can see C5b there, C6, C7, C8. And this is what's going on. If you go through this from the very beginning, so we have the antigen-antibody complex. This is the classical pathway. C1 is going to be activated by that. C2, C4, these guys are going to join together. And these guys are going to come over here, and C4 is going to split into C4A and C4B, and C4B is going to become part of that C5 convertase. C2B, C2A, C2B is going over there. Well, they've already got that shown right there. And then C2A, so C2A and C4B, the part that doesn't go there, there's some left over, are going to combine, and these are the guys that are going to split C3 into C3A and C3B. C3B is going to join with C4B and C2B to become the C5 convertase. C5 is going to split into an A and a B part, and C5B is going to be the part that helps to activate the membrane attack complex, the MAC, and that's going to punch the holes in the plasma membrane and the cell wall and cause these guys to lyse. So it looks very complicated, but it's really not, and I prefer to have things go kind of linear like that, but you can always redraw that to make it linear, or you can look for other diagrams or what have you that can show it to you in a better way if you need that. If not, that's fine too. Cytokines, proteins that we use for cells to communicate. These guys are released. They may cause other chemicals to be released or cause cells to do certain things. For example, we may cause the cells to start dividing rapidly, cause them to change in some way, might inhibit the same cell division, cause cells to destroy themselves, and also the chemotaxis, the the homing signal for the white cells. So we have, just as we do in hormones, we have autocrine, paracrine, and endocrine. These guys are going to secrete a cytokine that actually acts on themselves. There are cells that do that of the immune system. They release a cytokine that is intended to act on themselves. A lot of them are paracrines. They're going to release it, and it's going to be picked up by nearby cells. And then you have endocrine, in which it's going to be distant. Okay, guys, let's see what else we got. Okay, guys, going back to our cytokines, the three classes that we're going to talk about are interleukins, the chemokines, and then the interferons. Interleukins, remember that the white cells are called leukocytes, so you might pick up on the fact that interleukins are things that are produced by the white cells, and they are to communicate with the other white cells. In some cases, they'll also speak to other cells and cause them to perform functions. Okay, chemokines. A lot of these are the chemotactic factors like we were talking about with C5a and C3a. These guys are going to act as a homing beacon for the leukocytes to bring them to where the infection or what damage or inflammation is. Now, interferons, these guys, there's alpha, beta, and gamma. These type 1s, alpha and beta, these guys are released by cells that are already infected by a virus. So this cell has virus infection. He releases interferons. These neighboring cells are going to pick up on an interferon. So this neighboring cell, which is uninfected, is going to scale back on its protein production and going to destroy RNA, which means that the bacteria, if they do manage to get inside there, won't be able to reproduce. Cells that are already infected or in the beginning stages, this will induce them to undergo apoptosis to destroy the cell to get rid of the viruses. And also this will help to activate some of the immune cells, especially the cytotoxic T cells. So our cytokines are also going to start on our acute phase proteins. We have acute phase proteins, and one of these is C-reactive protein. You'll see this guy, abbreviated CRP, on the labs in the hospital. And the mannose binding lectin, MBL, see that abbreviated like that in the lab reports. These guys are going to act as opsonins. Now, opsonins, we haven't really discussed that, but opsonins are tags. So if this is a bad guy, no, that's not a bad guy. That's one of our cells. Here you go. So here's a bad guy, bacteria, whatever. When these guys are tagged, that tags them for destruction. That's one of the ways, well, that's one of the main ways that antibodies work as an opsonin. And we also know that these guys can activate the complement cascade through that lectin pathway. Some cytokines are going to bind to mast cells. This is a mast cell. Mast cells contain histamine, and histamine is pro-inflammatory. That's why people take antihistamines when they have allergies, because it's just an inappropriate immune response. Histamine receptors on a lot of different cells, and they're going to help trigger an inflammatory response. They also have things called leukotrienes, which are also going to be pro-inflammatory. Prostaglandins are mediators, and they are also going to be pro-inflammatory. And they're also going to kind of magnify the effects of the kinins and the histamines on the inflammatory. Hematopoiesis, again, you guys have had this in anatomy and physiology too, those of you who have had that, but we'll go through it in any case. There is the pluripotent stem cell in the blood or in the marrow, and from this it can differentiate into one of three. It can either become erythroid, myeloid, or lymphoid. The erythroid is going to become the erythrocytes, the red blood cells. The lymphoid is going to become a lymphocyte, and the lymphocytes themselves can differentiate into three different kinds, but we'll get there. The myeloid is going to become either the monocytes, which is a precursor to a macrophage, eosinophil, neutrophil, the basophil, or the platelets, which we need for our blood clotting. Okay. Defense components, we've talked about the leukocytes, the granulocytes. Those are the ones that have those large granules. Basophils, eosinophils, neutrophils, these guys stain blue. Basophils are very similar to the mast cells, and they have granules containing histamine. Eosinophils have this red-orange granules, and these guys are for parasitic worms. Very rare that you see eosinophilia in a patient. Neutrophils, these guys are going to be kind of clear. These are the main responding cell in the early stages of an infection. You'll see these guys, abbreviated PMN on the lab, polymorphonucleotide. Neutrophils are going to phagocytize bad guys, and diapedesis is the process of crawling out of a blood vessel into the tissue. Eosinophils, to a degree, they're really more for parasitic worms. The agranulocytes are the ones that don't appear to have these granules. You have the lymphocytes and the monocytes. The monocytes, when they leave the blood, they're going to become the macrophages. The macrophages are going to succeed the neutrophils in the tissues within 12 to 36 hours, and they're going to be the big phagocytes, and they're also going to be there to clean up any debris prior to repairing. Lymphocytes are adaptive immunity, except for one of the three, which is going to become the natural killer cell. He is part of the innate immunity. Okay, mast cells, mast cells, at one point they were thinking they were actually an activated form of basophil. Apparently they've decided that's not the case, but anyway. They have the histamines, allergic responses. These guys are all about pro-inflammatory. And of course the mast cells will be in the tissues, whereas the basophils are basically blood-borne. Natural killer cell. So when a cell gets infected by some sort of intracellular parasite, usually a virus, but it can be a bacteria, all cells naturally are going to take part of the proteins they make, and when they recycle them, they're going to put a piece of it out for the immune system to look at. We're going to have a type of T cell that's going to come around and check that and make sure that it's not foreign. If it's foreign, then this cell is presumptively infected and the T cell will kill it. However, there are some viruses which are very slick. They're not going to show anything on there. so the T cells aren't able to kill them. The natural killer cell will. If this guy does not show any type of protein on the outside, then the natural killer cell will release the chemicals that will cause this guy to go into apoptosis. Pattern recognition. They used to call this the non-specific immune system, but the truth of the matter is they are specific for certain things, but the adaptive immune system is highly specific for one single part of one single organism or virus. The innate system is specific for certain patterns, common patterns. Peptidoglycan. Which bacteria have peptidoglycan? Most of them do. So it's not really as specific as the adaptive, but it's more specific than we had initially thought. Flagellin is a protein that bacteria use in their flagella. LPS, oh, that starts everybody off. You remember that from the gram negatives. Lipopeptides, molecules that a lot of bacteria will express, and nucleic acid, viral DNA, or RNA. Our RNA, if you recall, is always single-stranded, but there is such a thing as double-stranded viral RNA. There is single-stranded viral DNA. Those things don't exist in us. So these structures, pathogen-associated molecular patterns, PAMPs. So these are just things that are common to a lot of bad guys that we can detect. How do we detect it? The TLRs, toll-like receptors. Phagocytes have these guys, and they will bind to various peptidoglycan, whatever, and they're going to initiate a response based on that. Once this binds, it's going to go to the DNA, and it's going to cause the DNA to produce a protein, which is going to cause the action. It can start an apoptosis, cause these guys to release pro-inflammatory mediators, or even stimulate the adaptive immune system to come in and get in on the act. Okay, pathogen degradation. We're going to use phagocytosis. These guys are basically going to engulf, swallow these guys. They do it like an endocytosis. You can see right there. We get a bubble, and then we have another bubble, a lysozyme with digestive enzymes. These guys merge. Those digestive enzymes come into contact with the pathogen, kill it, cut it into pieces, and those pieces are used to alert the adaptive immune system. Chemotaxis, that's Neisseria. Pseudopods are just extensions of the plasma membrane around and eventually enclosing them, and of course putting them inside a bubble of membrane inside. You guys can look at this: adherence, ingestion, fusion, killing, elimination. A lot of it is just exocytosed, but some of it is actually kept and on a molecule to show the adaptive. Inflammation is nonspecific. Redness, heat, swelling, and pain. Acute is what you get from sudden but short-lived events. Chronic, of course, is something entirely different. This is the thing that is thought to be involved in the development of different cancers. So acute inflammation helps you out. It's really important. We need to open up those blood vessels so the white cells can get out. And it also helps with tissue repair. And amongst the things that are coming out of the blood, besides the white cells, we won't see any red cells there typically, but we will see clotting factors, and we will kind of put a blood clot around the damaged area to keep the bad guys from spreading. Chronic inflammation is not a good thing. So there's a monocyte, there's the wall of a vein, or typically it's actually going to be a capillary. So the pro-inflammatory mediators are going to cause the muscles to open up, to have gaps between the cells, and these guys are going to squeeze out those gaps, and that's called diapedesis. Migration, since these guys are open, they're going to hello? Okay, so by dilating, we're going to increase the blood flow, which is going to increase the number of the white cells coming there. And these guys are also going to start producing some receptors that the white cells can use to slow themselves down. Think of it like rolling over Velcro and come to a stop. And once they come to a stop, that's that margination, is when these receptors, of course, at the edge. So the white cells, we want them over here and not in the center where there are no receptors. Once they squeeze out, they just follow those chemotactic molecules to where they are. We already talked about neutrophils first and monocytes. Tissue repair is the final stage of inflammation. Extra nutrients and oxygen, fibroblasts, macrophages, after they take care of the bad guys, are going to take care of any damaged structures so that they can be repaired. If it can't be fully repaired, then we're going to get scar tissue. Here's an overview. So the bacteria, this is like a splinter. Damaged cells are going to release prostaglandins, leukotrienes, and histamines. Prostaglandins are going to make the vessels, they're going to dilate the vessels. Blood flow is going to increase. Macrophages and neutrophils are going to squeeze out diapedesis. And the increased permeability is going to allow some of the antibiotic chemicals. And we already talked about the clotting factors. We're going to get swelling because of all the tissue buildup, and it's going to press on nerves and it's going to hurt. But part of that is to keep us from using something that's damaged. That's why it hurts when you stand on something that's inflamed. It's supposed to. The blood clot we talked about, more phagocytes, we're getting rid of the bacteria. The damaged tissue, the leukocytes, especially neutrophils, are going to form pus. Stem cells are going to repair the tissue, and then the blood clot is going to be absorbed, or the part of the blood clot that's actually over here on the surface of the skin eventually will become a scab and fall off. All right, guys, that has got it for this guy. We